Hey guys. So we went over DNA and DNA replication as a whole. So now we kind of got to go over the next steps. How do we get that replicated DNA into a freaking protein so it can literally do everything our body needs? So we're going to go over transcription and translation. So genes code for all of your genetic traits. Okay, so we're basically going to go over what do they code for, how does DNA code for cells and bodies, and how are these cells and bodies made from the instructions in DNA, because literally every single part of you is made from DNA. So this is, once again, the central dogma. DNA replicates itself, then it's transcribed to get RNA, and then it is translated into proteins. Okay, when and that protein is represented as a trait. When I first briefly described this to you guys, I said you have to think of it as like a language process. So we're in the terms of like DNA language, right? So we have to transcribe it or rewrite it in terms of RNA in order to translate it into the language of a protein with amino acids to get a specific trait. Okay, so metabolism is what taught us about genes. So there's an inheritance of metabolic diseases. So this suggested that genes were coded for by enzymes. So each disease, which is the phenotype because that's the physical representation, sorry guys, is caused by non-functional gene product. Okay, that's called signal transduction. So basically, if you don't have that enzyme, okay, so a non-functional gene, you can get certain diseases. So like Tay-Sachs, albinism, things like that. So this would be like the metabolic pathway. So we have an A here as the first step, and then enzyme 1. Basically, we're crossing that out as we keep going through. We went over signal transduction when we went over cell-to-cell communication. So we had these dudes, George Beadle and Edward Tatum. Okay, they discovered that genes act by regulating specific chemical events. Okay, so they had a one gene, one enzyme hypothesis. So this is what they did, is they looked at asexual spores, okay, and kind of put them through this process to test whether they could get a mutation present or not. Okay, so here by adding X-ray or UV light, they created mutations, okay? And then they had a positive control here and the negative control here. And then these were all of the experimentals. And then these were the amino acid supplements that they got, okay? They identified a mutation here in arginine. So basically what that means is that whatever enzyme took to get to arginine, that is where the mutation was. So one gene, one enzyme type of thing. Okay, so think of it this way. We have from the nucleus, we have our DNA, right? We have the DNA blueprint written out in instructions. Okay, then we have to write those instructions in a language that the ribosome will understand. So and then also remember, DNA cannot leave the nucleus. So since DNA cannot leave the nucleus, we need RNA, mRNA, which is basically DNA with a little mustache on, because it turns that T into a U. So since that happens, it kind of disguises itself so that way it can leave the nucleus and actually get translated into proteins. Okay, so we have to rewrite the DNA as RNA through transcription. Now we can leave the nucleus, find a nice little ribosome, so that way the ribosome can translate those instructions into proteins. And then from the protein, we get a specific trait. Okay, so think of it, like I said, all in terms of language. There's videos attached throughout here. So if you guys like need some extra like watching explanations and things like that, please, please, please do so. So RNA. We have to remember RNA is ribonucleic acid, whereas DNA is deoxyribonucleic acid. So RNA is single-stranded. It has the ribose sugar It has nitrogen-containing bases, just like DNA. But we have uracil, so U instead of thymine, T. Okay, so A matches with U. C still matches with G. We still have our phosphate group, but it is single-stranded. Okay, we have types of RNA. We have mRNA, tRNA, rRNA. mRNA stands for messenger RNA, so it's sending the message. tRNA is transfer RNA, and rRNA is ribosomal RNA. So here you can see mRNA is the actual template strand that we get and transcribe in from DNA. Our RNA is the ribosomal RNA, so it's the ribosome that's going to help translate. Okay, and tRNA is what transfers those 3 bases to become that amino acid sequence. So let's go through the process of transcription. So transcription is making mRNA complements, okay? So we have to disguise our DNA genetic code as RNA in order to get it to leave the nucleus. So it transcribes DNA strand, which is the template strand, okay? That's the antisense DNA. And then the untranscribed DNA strand is the coding or sense strand. We went over antisense versus sense strands in our DNA lecture. So the untranscribed RNA or DNA has the same sequence as RNA. Then you have the synthesis of complementary RNA strand, okay, at the transcription bubble. Because remember when we went over DNA replication, we had to create that little bubble so that way there's room for it to split up at the replication fork and actually copy. So the enzyme that helps us out is RNA polymerase 2, okay? Oh, sorry guys, haven't had enough coffee. Okay, so we have our template strand here. Okay, that's going to be our 3' to 5'. Okay, and then the coding strand is up top. We build RNA 5' to 3'. Now, if you remember, DNA has to be built 3' to 5'. RNA is opposite. It is built 5' to 3'. Okay, so here's kind of like protein size view. Okay, so here's the RNA polymerase. Okay, the promoter is the starting point. Okay, the promoter is the thing that helps them realize, okay, this is where we're going to start. We can get going. So RNA polymerase comes in and it's going to be transcribing this specific section here. Okay, so this is RNA polymerase, what it looks like as a protein here. There's 3 different types of RNA polymerases. Okay, you have one which only transcribes rRNA genes, so the genes to create that ribosomal RNA, and it makes ribosomes, obviously. Then we have RNA polymerase 2, which transcribes genes into the mRNA, which is transcription, the entire process of transcription. And then we have RNA polymerase 3, which only transcribes our tRNA genes, or transfer RNA genes. Okay, so these are huge, big daddy proteins here and enzymes, okay? So this is just kind of to give you kind of I guess, a visual of the size of RNA polymerase 2. This is a super not complicated way at all of showing you certain transcription factors that are activated, okay? So this is kind of breaking it down step by step in scientific jargon. Okay, so which gene is read? How do we know which gene to actually transcribe and start with? Like I said, there's this thing called the promoter region. That's the binding site before beginning of the gene. Okay, so the promoter region is basically the area that will tell you, hey, okay, we're about to get started with transcription. They call it the TATA box binding site. Okay, no, it's not tatas, okay? But it's just because there's thymine, adenine. So TATA, that's the box where you know it's a promoter. Okay, that's the binding site whenever that part of DNA is read. That's the binding site for RNA polymerase and the other transcription factors. Okay, Then the enhancer region is the binding site that's far upstream of a gene. Okay, so that just basically means that it turns the transcription on high. Okay. So what are transcription factors? Transcription factors are part of the initiation complex. So they bind to the promoter region, and they kind of like are a suite of proteins that bind to the DNA. So these are the proteins that are going to trigger the binding of RNA polymerase to DNA. So think of it as kind of like markers. It's like, hey, RNA polymerase, you going to be here, bro. Okay, so if they're there, they're the ones that can turn on or turn off transcription. Ooh, let me go back real quick. Okay, so here's where you can kind of see, you can see that this is like the binding site, okay, and so we have the transcription factor here that's like, yes, okay, I'm going to regulate whether or not this will get turned into a gene, and then it will know that you can begin to transcribe it. So you have to actually match the RNA bases to the DNA bases on one of the DNA strands. Just remember that instead of T, we're going to be matching with U, but T still matches with A. So we have A, U, C, A, G, and just so on and so forth. Okay, so this is obviously the 5' end for RNA to the 3'. Okay, so how does the transcription of a gene end? How do you know that this is the end of the gene? We can stop. We have row dependent, which means Rho or RHO, it's just a protein. Okay, so when that binds to mRNA, it releases it from the template. Okay, now when you have Rho independent, that's when you reach a guanine cytosine rich region, so an area with a bunch of guanine and cytosine, and poly A tail in DNA forms a break. Okay. So basically, if you have a Rho protein, then you're going to bind, it's going to bind to it, and it's going to basically tell mRNA, hey, bro, you good, you can go. But if you do not have that Rho protein where you have an area with a bunch of guanine and cytosine, then DNA forms a break. Now, eukaryotic genes have a bunch of junk, okay? They're not continuous. They're tons of little sections, okay? So you have parts called exons. Those are the real genes. Those are the coding expressed sequences, okay? So those are the sequences and the real parts of DNA that are actually expressed. So think exons expressed. Introns is all that other crap in between, okay? So introns in between. Okay. So we can have this thing called mRNA splicing. It's after transcription, we can make modifications. So after transcription, eukaryotic mRNA needs some work. Okay, it's not absolutely perfect. So you can have primary transcript, which is pre-mRNA. mRNA splicing, that's where it comes in and edits out the introns, which are the non-coding sequences, so all that nonsense and junk in between, okay? And then you can make mature mRNA transcript with a cap and tail. The cap and tail is kind of just like endpoints, just so you know, okay, this is the start, this is the end, we're capped, we're good. This section is a beautiful gene. So these lovely humans discovered exons and introns. Okay, so we had Richard Roberts and Philip Sharp that actually discovered what exons and introns were. Okay, and this is kind of like explaining the process that they went through to find it out. Okay, so RNA splicing, there's no room for mistakes because this is already a gene, like a shortened DNA sequence, okay? So they have to have a single base added or lost, which throws off the entire reading frame, okay? That's called a frame shift mutation. I'm going to keep going with this in the next one.